Mama ye banane mis zanan madiha mama
golden color
Zara madiha ye banan mamas mama bana ubas mien girl those are girl bu
वो मुझे गड़ी है ऊपर ये
तुझे जरूरत नहीं है बोल
किती पाहिजे 2 पाहिजे ना हो
Morning, Meera.
Morning, Dr. Bhushan. How are you?
Doing good, doing good. Yeah. Trying to guess how many participants are there for now.
Hello,
Hi,
Dr.
Oscar.
Bhushan.
How are you?
Good morning.
How are you doing?
Quite fine. Quite fine. Hi, Meera. Hello.
Good evening. How are you?
Quite fine. Quite fine. I hope that the people are joining still.
Yeah, I was trying to look how many are there. I was not able to get a sense for it. Yeah.
I will just check. I will just tell you the figure.
Okay.
Yeah. Now the 42. The toll is at 42 now.
Okay, that's good.
Yeah. Hello, Shailesh.
Morris has not been joined
हेलो
yet.
सर बोला सर जॉइन होता है सगळे
Yes, I joined.
Yeah.
Yes, still people are joining.
Okay, good.
I
We're
think
waiting for Dr. Morris to join us.
Morris, yeah.
So, Oscar, if you were supposed to go to Nagpur, how do you go from Satara to Nagpur?
So either by train or we usually take plane or bus, either of these. The usually 15 hours.
How long is
15
the
hours.
journey? 15 hours.
Yeah. Either by train or by bus.
Okay.
It's the, I would say, as per the distance is concerned, the longest college from Nagpur.
Oh, okay. Okay.
It's around 600 kilometers.
That's quite a lot. That's quite a lot. And probably there's no direct road, right? You have to go up and then go to the right. Is there something we can?
We have to come to Pune, and from Pune, either you take a plane or bus or train.
Okay, got it. Got it.
Yeah.
Now I can get the geography a little bit better. Yep. Yep.
It's nearly 60 kilometers from Pune on Pune-Bangalore highway.
How much is your enrollment at your undergraduate level? How many do you take every year?
Sir, 62.
62 students. And
Yeah.
for PG, how many do you do?
Two per department, so that total comes to around 36 for 18 departments.
Okay.
Yeah, and doctorate is in the 4 departments they are offering for doctorates also.
So in 4 departments, you're offering PhDs.
Yeah, PhDs.
Okay. So how far are you from Varnanagar?
Varnanagar is roughly 150 kilometers. It's on the Bangalore-Pune highway from Pune, just before Satara, then comes then Kolhapur.
Okay.
Morris is not exceeding.
Yeah, that's what I've been watching here for.
ये डॉग को पालते हैं ये डॉग को पालते हैं
Mira, would you mind calling Morris on his WhatsApp if you have his WhatsApp number, just to check? If not, I can do it.
किसको बचाते हैं
Don't have his WhatsApp number.
Okay, I'll do it. I'll take care of it. Yeah.
Thank you.
Hey, Morris, we're waiting for you. Are you difficulty in getting on? Yeah. Okay. Yeah, he sent you a new link this morning. So yeah, Dr. Zende sent us a new link, so you can probably click onto that. Okay. Okay. Okay, no worries. You want Mira to go first and then you want to set up yourself? Okay, okay, then let's do that. So it'll get you a little time to get organized, okay? Wonderful. Okay,
yeah
thanks. Yeah. He had some clash with timings, so he's going to join us. So Mira, would you mind, instead of Morris going first, would you mind going first, if that's okay with you?
Yeah, we can go ahead. There are some intersections with, those things Morris will cover at the end, but we can go ahead and do the.
Okay, okay. If it's okay with Dr. Zende and Dr. Wasker, if we can wait for a few more minutes to see if we can get Morris on the web, on our link here. So.
Naina. Hello. Sir, am I audible?
We can hear you loud and clear. Yeah.
Yeah. Thank you. Good morning, sir. You and my Indian participants, good evening to everyone. So I once again welcome sir for this fourth day of this international training program on introduction to bioinformatics, microbial omics. Today also we have with us the two eminent speakers today. Yesterday we have learned theoretical part from them, and it was very nicely they have put all the things regarding the shotgun metagenome sequencing and microbial community analysis, the concept and approaches both of the speakers. And Dr. Jairav sir, my teacher and philosopher, he has also moderated very nicely and communicated all the questions, answers from the participant to the speakers, and they are well explained to our PG and PhD students and our faculties of veterinary science across the country. So I welcome you everyone on my behalf and my NHP team and authorities of our university. And I request today also Dr. Jairav sir being as a chairman of this technical session, and Dr. Vaskar is co-chairman of this technical session, and Dr. Savani is a rapporteur for this technical session today. So now today we are going to see the demo from this Dr. Meera and Dr. Morris. So I hand over this now session to Dr. Jairav sir, please.
Thank you, Dr. Zende. Thank you for inviting us for the second phase of the Bioinformatics Symposium. Delighted to be here. We are waiting for Dr. Morris Biosinghe to join us. So I just spoke with him and he will be soon doing that. So in order to save some time, I just had a quick chat with Meera, and Meera has kindly agreed to start off the lecture today with some demonstration on some of the software that she plans to show to the people who are attending this symposium. So Meera, go ahead, it's all yours.
Thank you. Good morning, everyone. It's my pleasure to talk to you again this morning. Today I am going to do an overview of some of the tools which we use for microbial community profiling, mainly 16S amplicon sequencing or microbiome analysis. Thank you for sending us the survey as we mentioned yesterday. That gave us a gauge of what you have been used with, which softwares you have been using and things like that. Today, what I would like to go over is more like an R platform. I realize that many of you are not using our platform as much as command line options. So I want to introduce this to all of you. This is kind of a very beginner friendly tool to use with, and R is completely a software, free software program you can access. So that's why I want to bring this in, and the analysis we will be doing, it's a little bit different from the traditional OTU. I'm going to discuss more on that. Let me share my screens now. Okay, you're seeing my PowerPoint, correct?
Yep, it's clear.
Okay. So, what we are, what's our stage, or where we are setting up the stages? Let's think about an ecosystem where we are introducing a change, right? And we are looking for the microbial phenotypes in that ecosystem. And that's what we did with 16S rRNA gene sequencing. Now we are at the stage of looking at the data and do some bioinformatics and statistics. So what we are trying to look is the microbial composition or function in these different ecosystems. That's basically the stage where we start with. So we have definitely a question to ask, right? Then as a follow-up from what we discussed yesterday, we did the sequencing and we have the Fastq files, and we went through these three phases, like pre-processing of reads, OTU picking, and diversity measure analysis. So we will start with phase 1, phase 2, and follow through phase 3 in this demonstration session. So this is where we start with, right? We have a lot of Fastq files. Let's say it's a pair and V4 sequence reads. And after phase 1 and phase 2, this is what we end up with. We will get either an OTU table and a taxonomy table, from where we take it to the phase 3, where we look at diversity analysis. So in order to go through the phase 1 and phase 2, there are several clustering methods used, which is well described in the literature. For example, UParse, Med, or Chime, Mother, or Data2. Chime and Mother are mostly a command line based platforms. But recently, in 2018-19 Chime has been trying to develop a graphical user interface which is more accessible to beginners, so that they can go in and do their analysis on a web app. It's still under the construction phase or beta phase. So you can still go in and do it, but you need a virtual box to do it. It's not entirely on a web app yet. And Mother, this is something I have been using for long for OTU-based microbiome analysis. This is also a command line-based one, and they have very good reviews and descriptions out in the literature about how to use and what are the variations we can bring in Mother. But today, what I want to do show you is mostly a new program or new software called Data2. This uses an R platform. So Data2, the differences from Mother and Chime is Data2 is actually an algorithm which picks up amplicon sequence variants rather than traditional OTU.
Mira, are you moving your slides, or we are just set at the very...
Oh, okay.
We are not seeing it moving around, so I was wondering if...
Thank you for telling me that.
Yeah, sorry about that, but I should have told you a few minutes earlier. Yeah. Yeah, the same thing happened to Suresh a couple of days ago.
Okay, interesting. Okay, let me try one more time.
Yeah, give it a shot and see if you can move it around, so we'll tell you it's working. Okay, we see another screen up here.
Okay, that's enough. Yeah, let me stop share and do it again.
Using a
yeah, I was
using an Apple system, or
no, this is Windows.
Okay.
It was fine yesterday. I'm not sure why this is. This is presentation.
if you can go into the, rather than going to a full PowerPoint mode, see if you can change it while you are just on the standard screen. Yeah.
Okay. Here you go.
Okay, got it. Okay.
Yeah. Okay. So I went through this slide, and here we are now. Okay, I'll a little bit back up on this one.
Yeah, if you can back up from this.
Yeah.
Your cover
So
slide.
let's start from here again.
That'll be great. Yeah. We have seen this slide earlier, so this would be great. Yeah.
So yeah, this one was, we discussed about the 16S analysis workflow yesterday. As you remember, I was talking about different phases of processing the sequence rates out to the diversity measures. So the first one you see is the phase 1, where we start with quality control, trimming, parent stitching, and chimera detection. I told you we will be discussing it today. Yes, we are going to use different tools to get to the phase 1. Then the next phase is OTU picking, which is like operational taxonomic unit. This is a traditional way of analyzing the microbiome data, but instead of OTU, today we are going to discuss more on an amplicon sequence variant analysis, which outperforms OTU in many different ways. So today I will bring up the topic on amplicon sequence variant analysis and so on. And the third phase, we will use those data to evaluate the diversity measures. So from the sequencer, we get a lot of Fastq files. This is where we start with. So we have to go through the phase 1 and phase 2, and then we end up with something like a taxa count table and taxonomy table, which is what we use for the phase 3 analysis of diversity measures. There are several clustering methods which have been used for analyzing the microbiome data. For example, CHIME, Mother, or Data2. This CHIME and Mother are mainly command line based softwares. And recently, CHIME has came up with some kind of graphical user interface platforms, which is still under the beta form, or meaning construction form. So you really need a virtual box to do it at present, but they are moving on to a web app so that all the beginners can start using it and can analyze the data on a web-based CHIME platform. Both this CHIME and Mother traditionally uses operational taxonomic units. But the one which I am going to discuss today is a platform or algorithm called Data2, which uses amplicon sequence variants and uses an R platform. And there are well-developed tutorials available. At the end of this presentation, I will share with you some of the resources which you can use for your study. So you might have remembering this slide also I discussed yesterday. So what essentially it's I want to tell you here is, what is the difference between OTU and ASV or amplicon sequence variants? In an environmental sample, say if you have 4 unique sequences, during amplification, some amplification error arise and create similar sequences but slightly different, and it introduces some noise into our data set. So traditionally what happens is, it clump up everything into OTUs, usually at a 97% threshold. So then thereby grouping these clusters into sequence similarity. So you can see an inherent limitation to that. Although this green and red are slightly different, they are grouped into one OTU. However, in the case of Data2, what it tries to do is to resolve some of these errors. It's simply based on sample inference, meaning based on your sequences, quality control, abundance, it makes a call whether the sequence is real or not, and then fine-tune your answer. So many ways Data2 outperforms the OTU picking algorithms. So as I mentioned before, this is the platform we are going to use. I'm not sure how many of you are using our platform, but my recommendation is it's a highly powerful statistical platform. It's free to use. There are a lot of tutorials available on different packages for microbiome analysis, metagenomics, and so on. So it's always, my recommendation is to start working on some of the R packages, which will give you a lot of flexibility in describing your data. So Data2 is actually an R-based platform. So we'll be using this R platform today, and I'm going to demonstrate few of the tools which we use with Data2. So if you're interested to download the R, this is where you can go and download the R. There are some graphical user interface apps which can link this R, which is which is called R Studio, and so R Studio will give you a clean screen to start working with. We will go through all these things down the road. So as a starting point for Data2, this workflow in fact assumes that your sequencing data meets certain criteria. For example, the samples needs to be multiplexed or split into individual per samples Fastq file. If your sequencing facility give you a long Fastq file with all the data included in it, I would recommend using some softwares like iTemp. which can help you to demultiplex your data into individual Fastq files. The other thing you need to consider is you need to remove all the barcodes and adapters before we put the Fastq files into the adapter. So you can use, as Dr. Morris mentioned yesterday, either Qtadapt or Trimmomatic, and you can those ones to remove the barcode or adapters. And the third important point to consider here is if it's a paired and sequencing data, the forward and reverse Fastq files contains reads which should be matched in the order. So the data today we are going to work with is a paired and 250 base pair reads generated on an Illumina MySeq, and it's an amplicons of V4 hypervariable region of 16S rRNA, and it's amplified using the standard primers for V4, which is 515F and 806R primers. These primers are well described in the Earth Microbiome Project. So if you search for Earth Microbiome Project, they have a list of primers you can use for different variable regions. And today the data we are going to use are the gut samples collected longitudinally from mouse host feeding. So this is the data set we are going to use today to demonstrate the data to workflow. So basically in a data to workflow, these are the different steps. One, it starts with pre-processing the sequences. It goes into filter and trimming. We will learn more about error rates in our sequences. We then do the algorithmic sample inference to create the amplicon sequence variants. The forward and reverse reads will be merged to get the full information out. Then we remove the chimeras, assign the taxonomy, and we do some analysis and visualization. This is a picture of how chimeras are created. This happens when their primers are not removed properly from the FASTQs, or the PCR has some imperfections while it has been pursued out. So there is a biological X sequence or Y sequence, and they join together, and they are chimeras. They are not exactly the sequences which we are looking for. So we need to remove such kind of noises and clean it up before we get to the ASV table or taxonomy table to start analyzing with. So this is an overview. Now what I want to do is I want you to walk you through the R platform using the data to software, and I want to use the data to show you how different things we can perform in the R studio. Okay, let me share this. Okay. Can you all see my, oh, you're seeing my PowerPoint or R?
Yeah, Mira, we can see your script files up here.
Okay, sounds good. Okay, so this is basically how the R platform looks. I'm sure some of you may be aware. This is called console. This is where we put our scripts, and then you have an environment space, and there are some file space. This is how it looks like in an R studio, and we are going to use this data to package to do our sample analysis. So for that, what we want to do first is we need to install the data to, and this is the command we have to use to install it. So as I mentioned earlier, I can give you a good resource where you can go in and try this tutorial by yourself. This is just, I'm going to demonstrate it now. And when you do it, if you have any questions, feel free to reach out and ask if you have any. So then, so first we need to install this package. Once we install the package, we want to load the package onto the system. So we are now loading the package. So once the package is loaded, then we want to tell the software where our files are. So we need to tell the path where to go to get our files. So today I'm going to use 6 samples, 3 from a pre-weaning stage of the mice and 3 from the post-weaning stage. So I'm only going to use 6 of them, just the sake of time. And if you have more sequences, it needs more memory, more computational time, and more RAM. So just to get things done on time, I'm just going to use 6, but you can use as many samples as you want. So I'm going to get my path in, and then I'm going to ask to list the files. So when you list the files, you can see here down the different files I have here. This is the different Fastq files I have here. I also have some reference databases downloaded. We'll get to it, but I can see my files and I am on right path. That's what it means to. So once we have the files in, what we want to do next is we want to sort the names of the Fastq files. Why we want to do it is we want to make sure we have the R1 and R2, the reverse and the forward reads there, and they are matched. That's the next step we want to do in this process. So I'm going to sort my R1 and then R2. And I will strip off the names to make sure that I have both R1 and R2 there. Once I figure out the R1 and R2 files are created, then the next step is to see what's the quality of these sequences. I'm just going to look at the first 2 forward sequences and see how it looks like. So in order to do that, I run this command here, and I'm waiting for the quality pictures. So you can see I'm looking at 2 different samples of R1s, and this green line indicates the average quality of the sequences there. So you can see it's almost, it's a 250 base pair reads, and almost all of the reads have around 30 quality for QC quality score. This is good, in fact. So this forward primers are looking pretty good. Forward reads are looking pretty good. And there is no need to trim much of these out, but as a sake of to avoid uncontrolled any sequencing errors, what we are going to do is we can trim off like 10 sequences just to avoid any unwanted errors. So this is good to go. So looking at this, we can say that the sequence, provided the sequencing run, all the samples were run on the same sequencing run, probably the forward reads looks much better. Now, so now we need to see what is happening with the reverse primers, reverse sequences. So let's look at it. You can see that the reverse reads are not that good as the forward reads, but this is okay. This is common with Illumina sequencing. Mostly the reverse reverse reads are not as good as the forward reads. So where the quality drops off here is approximately at 160. You can see that the quality is kind of dropping off. So 160 base pairs, the quality is kind of dropping off below 30. So I'm going to trim, we can trim it at this level. And one thing you need to remember is that we have a 250 base pair paired, so we have enough to cover both regions. If you remember, the V4 region is just 254 base pairs in size. So both forward and reverse, when we overlap, we have enough of coverage. So it's okay if we even if trim off 160 with 160 here in the reverse read, we have enough overlap to get the full V4 sequence. But if you're trying to use any other regions like V1, V1 or V2 or V3, you need to consider what kind of reads you are using is a 150 base pair or 250 base pair, whether the overlapping is good or not. If in that case, you need to come up with a threshold, how much you can trim off before you jump in and do it. So here we are good to go because just like V4 and we have enough coverage with this forward and reverse reads. So, so that's what we are going to do next. We are going to filter and trim these files. And I'm going to create a new folder to put these files separately so we have a clean reads to do the analysis. So I'm going to create a folder naming filtered to put all my filtered F forwards and filtered reverses, and then all the sample names too. Okay. So now let's look. Now let's filter these reads. As I mentioned before, you can see few commands here. So what I am trying to say is I am asking the software to truncate the sequences. The forward reads up to 240, keep up to 240, and the reverse reads keep up to 160. And I'm telling them not to take any ends, and maximum errors is 2 into like 2 error points in forward and 2 error points in reverse. We are also asking to keep the quality as such and then true. These are the different commands which we are trying to say through this. Most importantly, look at what we are saying for truncating. So with this, I'm going to truncate the reads. And I'm going to look at it how it looks. I'm getting some errors here. It's still running, so you need to wait for a few minutes. Yeah. So now you can see that here we started with 7,793 reads. And now we have 7,113 after all the quality trimming and filtering. In the first one, similarly on the second, we had 5,869 reads. Now we have good quality 5,299 reads. Similarly, you can see through the different forward reads how the trimming has worked. Then, unlike the operational taxonomic units, data2 takes into account of the errors in each sequences. This is good, in fact. So we can average the how much error we are seeing in the sequences. This helps the algorithm to actually pick out the exact sequences, not any sequencing errors or PCR primer error, PCR errors or any other spurious sequences. So in order to know what the error rate for each sequence, this algorithm actually look for the errors in each reads. So that's what we are going to learn next with these commands. We are going to learn about the errors here. And we can actually plot the errors and see how much the error rates looks like. I will stop in between. I just want to ask, are you all following what I am saying, or I'm too fast from the participants? I would like to get some feedback if you can put in the chat box.
Yes, madam, we are following.
Okay. So let me know if I'm too fast or if you're not following. I can stop in between and pace it down. I know this is kind of lot of command lines and things. So if anything you feel like needs to be explained again, please let me know. Okay. So after learning about the errors, we are going to look at how to plot these errors. That's what we are trying to do now. It's a little slow than just bear with me for a minute. It's showing a little slow. Yeah. So this is how the errors are plotted. So this black line follows what the actual sequences are, and we want all the dots to be along this line. So that's what observed sequences, sequence outputs, and this red line shows the fit. So this is good. Actually, most of the dots follows the black line, which indicates that the sequences are pretty good with less amount of sequencing errors. Similar way, we can also plot for the forward reads as well. So once we figure out what is the quality of the sequence we are looking at and what are the error rates, then we can go ahead and start doing the data to algorithm as such, which is actually the sample inference. So with this filtered reads, taking into account of this error measures, now we are going to ask the algorithm to actually pull out all the sequence variants or the amplicon sequence variants, which are exact sequences which we require to map to the reference database. So if you see here, that's what I'm going to do first. For the forward reads, I am asking to do the sample inference and give me the sequence variants. And then I'm going to ask the reverse reads to give me the sequence variants. So here in the data too, unlike time or mother, we are not merging the reads prior to all this quality control and trimming. Here we do both in separate ways, and we merge them before we create the amplicon sequence variants. So we will get information from both reverse and forward reads, which is kind of a powerful way to look at the errors and any other sequencing biases. So you can see here, among the 7,113 filter reads we started with, we have 1,979 unique sequences in sample 1. Similarly, you can see what's the stats for your unique sequences in all your samples for both forward and reverse. And from these unique sequences, actually we got, for example, 128 sequence variants in the sample 1. So that's kind of an OTU. You can relate that sequence variants to OTU. So that sample had that many OTUs in the data set. So just if you are more familiar with operational taxonomic units, you can always think about the sequence variants as OTUs for the time being to visualize what we are trying to say. Okay, so we got some sequence variants out of each forward and reverse reads. Now we want to merge them to get the complete information out of it. So we are asking to merge all the filtered data to analyzed data set to merge according to their R1 and R2 orientations. So you can see that we have this pair read successfully merged, and we have the inputs now and the outputs. Sorry. So this is how it looks like. These are the sequence variants. So instead of a name for OTU, what we get is a sequence, a long string of sequence. So yesterday we had a question about what happens if something is not in the reference. So in this case, once these sequence variants, we get the sequence variants, and once we align that to the reference database and it comes out as unclassified or undetermined, you can always take the sequence variants, take it and blast it to the NCBI blast platform, and you can see where it blast actually and what is the closest match to this. So this is also a way to understand what else is in the, is there any unknown or unclassified organisms which is present in your data set. We can closely align to some of the related species or genus and can get some more information. So this is also a good way to look at. And you also want to remember that these are not clustered ones like OTUs. These are each sequence variants from the sample. So each sequence variant we are going to map to the references to get the information on what is that, and the numbers they will get from that instead of clustering. So yeah, so we have our sequences now. So now I'm asking to create a table like an OTU table so that we can see how many we are having in each sample. So you can see that in all the 6 samples, altogether we have 203 Amplicon sequence variants, or just like operational taxonomic units. And it's also important to note that our sequences are pretty good in length. Most of them comes under the range of 250 through 254, which actually good, which is what we are expecting to see, because 254 is the normal, the size of the V4 regions, and we got the full length of V4 in all of the sequences. This is pretty good with all the quality filtering and error matching we did. So now the next step is to remove chimeras. So those ones from which we got, we now need to, I want to remove all the chimeras present in this data set. That's the next step we are going to do. So we asked them to remove all the chimeras from the data set, and we identified 28 out of 203 input variant sequences. This is not bad because it's actually only account for the 3% of the merge sequence rate. That's not that bad. If you see more than 14 or like 10% or 14% or 15%, then you have to go back and look whether you have actually taken off all your primer sequences and adapters out, and want to make sure that you are really looking at the sequences which you are intending to. It's not like spurious results. 3% is okay because there can be Bayesian biases and stuff. So that's something you need to take care of while you perform all these analysis. Okay, so now we have the sequence file, and I just want to show you how your OTU table or ASV table looks like. Let me show you my CSV file on that. So I asked to write a CSV from this sequence file so that we can see what we are looking at. Can you all see my Excel sheet here?
Yeah, Meera, we can see it.
Okay, so these are the sample IDs. So as I mentioned before, we have 6 samples, and instead of OTU 1, 2, 3, you are seeing a long string of sequences or the nucleotide bases, which is each one is an ASV. So what you can do is from now on, you can rename it as ASV1, ASV2, ASV3, and have the sequences as a backup, as a separate file. So name it this one as ASV1 and copy it to a sheet, the sequence identity. So it will be easier for the downstream processes. But what I want to show you here is as similar to OTUs, you will see like 579 of these sequences, 405 of these in the F3D1, and the numbers. The number of ASVs, ASV1, for example, in these samples. This is what we get from this after we process through the data too. Also, the next step is to see which taxa they belongs to. For that, we need to have taxonomy assigned. So for that, I'm using Silva database. Let me see whether I can share one more screen of mine. Can you see my R file too?
No, Mira, not at this time.
Okay. Now you can probably, right?
Yeah, now I can see, now we can see your R file. Yeah.
Okay, so now I'm going to assign taxa to these sequences using Silva database. As I described yesterday, there are different bacterial databases available. You can use Green Genes or Silva. Here I am using Silva. With my experience, it seems like Silva is getting updated every now and then, and we get new information from it. And also now we can do species assignment to it. It's not perfect because V4 region is small and species assignment is not always complete, but at least we can get to the stage of genus. So that's what I'm going to do here. I am going to assign taxa to the sequences. I'm going to print this into a CSV file again, and let's look at what taxa are we seeing. Now see, these are the different phylogen class, like the taxonomic assignments we got for this is the ASV1 or Amplicon sequence variant 1, and it belongs to the kingdom bacteria, bacteroides. You can see a lot of sorry, let's see. Bacteroidales here. This is mainly this is actually a mouth gut sample, so majority of them were bacteroidals, and you can get some information about the genus, but not much about the species. So I only did the normal Silva database. I did not try the species assignment yet, but you can try doing species assignment with a data set as well.
Mira, I have a question for you.
Mm-hmm.
Yesterday did a fantastic job of explaining the relationship of diversity within a given set of data points that you're collecting, right? Looked about it. Now, when you present this data here, these findings here on this Excel sheet, will you be able to go and calculate the Simpsons index and the other index based on this?
Yes.
And how
yes.
confident are you with the data points that you will be coming as it comes to developing a Simpson index for this one?
Yes, that's what I know that's what I'm coming to. So the next one,
okay. Okay, great. Yep.
This is just the first step. These are the two tables we want. One is this ASV table, and the other one is a taxonomic table to do any type of analysis down the road. So this is what we needed first. Once we have these in hand, then we want to import these data into a package called PhyloSeq, and then we will be able to visualize the alpha diversity and also the beta diversity, and also look at the different abundant taxa in each of the samples. Let me show you with the sake of interest. It's slower than I expected, but it's getting there. Okay, so what I have been trying to do is I was trying to put this data set into a PhyloSeq object. PhyloSeq is the package which we use to do the downstream analysis. This is not working. Sorry, there is something. I will show you, for the sake of time, I'm going to show you the workflow. So this is the tutorial I was mentioning. So far, whatever I explained to you is the same in here. I wanted to demonstrate in the art, but it's showing some errors. So I just want to show you briefly on how we can get to alpha diversity and beta diversity. So you remember we have the taxa file made. That's what they are using to build this PhyloSeq object. And with this PhyloSeq object, we are going to plot the alpha diversity here. So you can see that with the taxa table we created and the number of amplicon sequence variants, they were able to calculate the Shannon and Simpson. So I had 6 samples. Here they had 9 samples. You can see that all the early ones were had an alpha diversity of almost 4, but the late ones had an alpha Shannon index of 3.5 to 3.75. So with this data which we created earlier, that taxa table actually helped us to create this Shannon and Simpson index based on the number of ASVs we found. Similarly, we can also use this to create this beta diversity plots. If you remember, we are using ordination plots or NMDS. So we create distance matrix by giving this command to use it with Bray NDS. And we are seeing that with the distance matrix, the clustering is changed. So all the late ones are clustered together and all those samples which are from pre-weaning are clustered together. So early and late are the 2 different sets of data set we had. So this is also from the same taxa table we created. What they do is they make a distance matrix between the samples based on the abundance and the presence of different amplicon sequence variants. It's very similar to what you're used with like OTUs, but we are using here amplicon sequence variants. Similarly, now the next step is what different phylum or genus or family members are differentially abundant in different ones. You can see here with this commands and using the same taxa table, the taxa count numbers, we are looking at how many different Bacteroideaceae here, how many different Lachnospiraceae here. So you can see with different colors what is the relative abundance of each of these family members in different samples. So this way you can also see which group of members has increasingly expressed in one of the groups, whether early or late. So this is how we come to the diversity analysis. This is what I wanted to demonstrate on the R, but somehow it's not showing or it's slowing up. If you have any questions, please let me know.
Do you want to reload your R by any chance if it is spooling somewhere in between? So
Yeah.
it's okay. No worries. Yeah.
Yeah, once after Morris' presentation, I will try to pull up again and show you how our taxa table converted into the alpha diversity measures and beta diversity. But I'm happy to answer any questions, and if you have any doubts on how we came to the stage of diversity analysis, please let me know. We can discuss more.
So let's take a few questions for now, and we'll wait for Morris to set up and join us. Yeah.
Ma'am, I have a question.
Sure.
That's regarding your rules of filtering. You say you apply certain filters, right? Before you actually go in to start analyzing your sequences. How much does the filtering affect your final data? What is the bias that personal bias that could come in during your filtering process? I mean, or rather, I would ask like this: how do you set your filtering process?
Yeah, good question, Meera. So the answer to this is we don't do it by personal preference or personal bias, but when we look at the first data set, right, what we are seeing is what's the quality scores. That's what our basis for trimming. But provided that we have already tried to remove all the adapters and barcodes before put this data into data 2, once we have that clean adapter removed sequences, then we go back and check again how the quality looks like. If the average quality falls below 30, that's what I use to do as a trimming filter base. So for the forward reads, which you have seen is they are almost like all of them were 30. So I did not do much of the filtering or much of the trimming there. But for the reverse reads, the quality was dropping by almost around 160 base pairs. So I asked the program to keep all the 160 base pairs and then discard the bad quality base pairs down the reads. That's what I use for filtering. And then the rest of the trimming and everything, or error correction and everything is done by algorithm itself. So then I am asking the algorithm to look for errors in the sequence. And I'm asking, like, if you are seeing error, they do a predicted and observed value kind of algorithm. That's what you saw in the error plots. So they see whether it fits best. If it fits best, they pass through this error correction, and then if they are not, they will show a flag showing that some of the reads are not good enough or the errors are high, so they cannot use these reads up. But we did not get anything like that flags with this data set, but that's what happens. So that's also not I am personally or any of you are putting any threshold on it. They are calculating with this given data set as a training set, and they look for predicted and observed values and figure out whether the error rates are good to pass this filter. Did I answer your question?
I think Meera is muted. So
Yeah. Can you say that 30 is a good start either ways, forward and reverse?
Yes.
Okay. Yeah.
Yeah.
Okay. Thank you, ma'am.
Also, one thing you need to consider is what region are you looking at? I'm just talking about V4 region now, but there are some regions which has very less overlap. In that case, you need to consider whether if you trim down to like, let's say 160, whether you get enough overlapping. That also you need to consider. Otherwise, those reads will be excluded from your analysis.
Okay. Thank you.
One of the things I would do, Mira, here is take a known set of sequences that are already deposited in the PubMed database and go and experiment with them. And the endpoint measurements you really need to be looking at is, do you change the species of the organism by your trimming? Change the species of the organism by your. So if in the event, if you are seeing where your trimming is being affected, that's one of the learning processes which I usually tell my students to do, is to take some of these sequences off from PubMed and play around with it in order to get an appreciation as to how to eliminate some of the inbuilt biases and the external biases that may creep in when you start playing around with the sequences at hand.
Okay, sir. I get you. Thanks.
I understand this is more like an mathematical algorithms, and it's like a little bit more of a boring subject than what we discussed yesterday. But once you get the feel of it, once you start trying out, as Dr. Bhushan mentioned, you can make the decisions on how to plot your data, how to analyze your data or group them and things like that. So data too is very powerful, and many of the stuffs we are not making any bias on ourselves. So it has the algorithm already built to take into account of different errors which error sequences can have. So that's good.
Any more questions for Mira?
Yes, sir. Good evening, sir. Good evening, madam. Good morning.
Good morning.
Yep.
Yeah. Sir, since yesterday I'm hearing is very excellent presentations from madam and Maurice also. Yeah, madam, I'm Dr. Vilas Vaidya and working as assistant professor in the Department of Veterinary Public Health, Mumbai Veterinary College. And in our department, what we are doing, so we used to send the samples for the sequencing for to do that from other agencies, from private agencies. And many a time we got the accession numbers also from the NCBIs. We used to submit to the NCBIs and we got the accession numbers, because in our institute, we don't have the facility. But only one thing is I want to know, we want to set up this facility at our institute, to establish such facility at our institute. And only the thing is that only the sequencer or analyzer is required will suffice the purpose, or what other facilities are required, facilities are required, and what will be the cost of this sequencer, tentative budget will require, so that we can establish this facility at in our institute, madam, please.
Bhushan, do you want to support establishing the facility? I can bring them up to the analysis page.
Okay. Yeah, I knew this was a loaded question when Dr. Vaidya started off. So the whole purpose when we look at doing genomic and bioinformatics, if you're trying to set up a lab,
Yes.
We really look at it. What kind of service are you planning to provide? Is it for research? Is it for actually providing services to your client? What is your endpoint? Okay.
Yeah.
So it largely depends.
Yeah.
Go ahead. It's for research, right? So are you expecting a large flow of samples, say 30 samples, 40 samples, 50 samples a month? Or you're expecting one a week or two a month? What is your pattern? That's the first thing I'd like to sit and figure out from you and Dr. Zende and say, tell me how many samples are you going to be doing? Okay, so let's assume you're going to be doing 50 sequences every month. Okay, let's step around. So the first thing you will require is, in terms of equipment, is getting a small microbiology lab or a virology lab set up in place in order to get your pathogen to a fairly clean preparation for you. So that I believe you already have. So that's not an issue. Okay. So the next stage is to where we say is preparing the libraries or library preparations. So you require an area to do where you do your library preps. So many a times when we do our library preps, we go with the kit that is being recommended by the manufacturer of the instrument. For example, if I'm going to be using Illumina, and if I'm going to be using the Minisec platform or the NextSec platform, I usually will go and utilize the kits that is recommended by the manufacturer. Okay. The next important thing is yes, you get a good amount of DNA, but that doesn't tell you your DNA is good. You need to really know the quality of DNA. What you throw in the machine is absolutely important. Otherwise, you're going to be messing up and spoiling the entire sequencing process. The first and the foremost is to be confident of the quality of the DNA you're putting into the material. Okay. So here we really went and put a lot of efforts in order to see how good our quality is. So we purchased a tape station in order to go and look at the quality of DNA. Usually we used to do it by qubit and nanodrop systems, but we really were not confident that we are putting real quality DNA into our machine. So after we bought in the tape station, we have very little errors in terms of the sequencing process and the data output. So the first critical step is to get the best quality DNA and the reads. Also, it depends on the length of the reads you want your machine is able to do. Okay. Sometimes you would like to do long reads, sometimes you'd like to do short reads. So that's where I would leave it to the instructor. So typically we would like to see around 400 to 600 base pairs before we shear and put it onto the machine. And next is many a times we can go with low, intermediate and high depth of sequencing. Where do you would like to do? Because if you go with intermediate and low, you can throw a lot more DNA sequences, a lot more samples. If you go with high, you can throw in fewer and it takes longer time. So where do you optimize at that point in time becomes very important at this point in the process. Okay. So say you've got 50 isolates. Usually in a mid-level sequencing, I can put close to about 25 to 30. And it all depends on your lab manager who has the nose. I can say, I can even get away with 35. I know based on the reads I'm seeing here, I can put 35 samples with the barcodes and with the tags on it and ready to go. So experience becomes very important at the stage when you're creating your library preps and loading onto the machine. And once the data comes out of the machine is where we then start utilizing some of the softwares and understand what's the quality of the reads that are coming through. Is it amenable for us to do bioinformatic analysis? That's the next stage. And once you feel confident that your good quality reads are coming out, and the machine will tell you. For example, we have 3 different platforms we use in our sequencing lab. One is the Minisec, we have the NextSec, and we also use MinION. So we have 3 different platforms that we tend to use. But our true workhorse in our lab is the Minisec. So we were earlier with the Ion Torrent platforms. We switched away from Ion Torrent to the Illumina so that we are compatible with the other diagnostic labs within the Pennsylvania system. So all of us are on the Illumina database. We get fantastic support from the Illumina company. They're always there. Even if you have a question, you pick up and you call them. They are at your call. So also they are very helpful. They are proactive. If you're doing customizing of your reads, if you're doing customizing of your projects, they will be able to come and help you out. So again, it depends on where you would like to go, but we as the Penn State Animal Diagnostic Lab are extremely happy with the service we are getting from Illumina. Okay, so I would initially explore the different types of Illumina platform. The ideal workhorse would be to go with a Minisec to start off with. Probably it ranges anywhere between $60,000 to $75,000. That should be good enough to get you a lot of the day-to-day sequencing done. I would highly encourage you to buy tape station because that will reduce your errors that are associated with making your library preps. Because if you don't do a good job of your initial DNA, you're screwed up, totally screwed up. You don't even need to be doing it. So that's what the lessons learned is: get a good. If you can't afford an tape station, definitely go with the different levels of the qubit modules that are now being made available to apply to the genomics process. Okay. So another thing is training. It's not that Mira was able to just step in and start using the Data 2 program. She has spent years of time trying to analyze and wrap her minds around it and how to do it. So also with Morris, it's very important is, yes, you got the sequences. Ultimately, how do you make meaning out of it? Right? So that's where I really look at it as a three-part system. First part is the pre-part, the middle part, and the later part. We all tend to focus upon DNA extraction, sequencing, but we forget we need quality DNA to go in. We need clean isolates to go in. And then when you get the data, you should know how to analyze it. So all three aspects of the pipeline is very important in order to have a successful sequencing facility at your place. But the starting point, again, is the volume. If you're doing few samples a month, don't even bother to set it up. Send it off somewhere because the cost-wise, it becomes much cheaper. Okay, so all that you do is invest in the bioinformatics end of the pipeline, and then you're all good to go. So that's what would be my 15 minutes of spiel for you at this point in time. Does that help?
Thank you so much, sir. Yes, sir.
Mira, you're our sequencing guru. Tell us more.
No, what Dr. Bhushan mentioned is what I had to say. But with the bioinformatics, if you have few samples, just send it out, and you can train your students on bioinformatic portion of it. Just like let them try and error different pipelines, workflows. It needs some training, and then that's where you can put your effort in if you don't want to continuously sequence, if you don't have many samples to sequence every month. So yeah, I echo what Dr. Bhushan said.
Okay, any more questions from Mira?
I have added a link to the chat for everyone just to follow. There's a tutorial for Data 2. It's easy to follow. Just go through it. It has a sample set which you can start training on yourself and start using our platform. It's a very powerful platform. It's free to use. And once you get the hang of the packages, you can definitely use for your day-to-day statistics as well.
Morning, Morris, or good afternoon, Morris. How are you doing?
Good afternoon, Dr. Bhushan. I'm doing well.
Mira, thank you so much for your talk. Appreciate it. I think we'll switch gears to Morris and then come back to both of you in the later half. Is that okay?
Yep.
Yeah, sure.
Okay, Morris, go ahead. Take over.
Okay, so I think I will stop my video and share my screen. It's going to be an empty screen for now. I hope you can see my screen now.
Yeah, we can see it, Morris.
Okay, perfect. So today I had two two issues. But first of all, I want to thank Dr. Mira for her presentation, because some of the things she presented are overlapping with what I will be presenting, which means I will have to go through some of those very fast. So just to let you know that usually metagenomic analysis workshops are 1 week long, usually, to go through all the steps and have an idea of how everything runs. The setup of the computer itself will take like an entire day, let alone analysis. So what I will do is just go over what we said yesterday, what we presented yesterday, and then go in the steps and see what are the tools which are available to us and how they're used. And then one issue, one problem I had, you've seen that I connected a little bit later, it's because this bioinformatics goes with computer compatibilities. There are computers that are better than others in performing particular jobs. In my case, I use a Linux or Linux Ubuntu computer, which for some reason was not accepting to connect with WebEx. So I tried and kept trying. I couldn't get it connected to it, which is difficult for me because it will be difficult to demonstrate all the work I did. So I had to copy and a few of the output files that I will share with you. But one thing that I want to just let you know is that even after this workshop, it will be a good idea for anyone who is interested to do analysis of metagenomics to look for a tutorial like this. This is called the metagenomic tutorial using this particular software that we talked about yesterday. And what I will do today is to show you just what are the commands, what they do, and then share with you what are the results that we would expect from that kind of analysis. So, and another thing, I am preparing, so I started, I'm running, even my computer right now is running some of the data that I download from the NCBI database. And I'm writing right now a tutorial like this one, which is specific for this particular workshop, in which we will be doing step by step on what you would expect if you were doing a real life experiment. Then at the end, I will share with you each one of these steps, what are the output in a separate file that I will share with Dr. Zende. Hopefully he will be able to share with all the participants. Now, so in this particular tutorial, they used a set of samples, which I'm going to present. And those samples, the data are coming from this paper. So the paper was comparing or looking at the abundance of oral microbiota associated with oral cancer. And whatever thing you're looking at, whether it's cancer, whether you're looking at antimicrobial resistance in different animals, or the microbial communities in chickens versus the community in pigs, it will be the same. It will be the same process. What's going to be different is how you interpret the data and what they mean in real life. So this is the data from this paper, and they are explained very easily where you can get the data on NCBI database. Now, so coming back to the tutorial, so they show you what are the tools you need to have. So I'm going to show you how to use human 2. It does. So the name is misleading. It has nothing to do with humans like a human. As you can see, it's double N at the end. This is just a tool to do analysis, to do the metagenomic functional profiling. And then Metaflan 2, actually Metaflan candidate, because this was written in 2017. Right now it's not 2 anymore, it's 3, which has a few differences in how it runs, but all the principles are the same. And then STAMP, which is used for visualization, but we're not going to use STAMP because, I mean, we can use STAMP, but the computer I'm using now doesn't have it installed because it has different requirements, which does not allow it to get installed on my computer. Now, so what they did in this tutorial is they took all those samples and then reduced them because if you use the entire data set, probably your computer will not be able to run it if it's not a high-end, high capacity computer. But the link that is here, if you just type into the shell, what I call type into the shell, if you are using a Mac computer, or if you're using a Linux computer, you will have something that we call a terminal. It looks like this. It looks like this. So what you do is you copy the entire command and you paste it into the shell, and you do the work. Like I told you, here it doesn't work on mine right now because I'm using a different computer that I use for data analysis. But after you do that, just after you copy and paste, what you get is what I have in a folder, in a separate folder here. It's called MGS tutorial. Then you get a number of folders. Clicking on one on a file like this shows you what we have in the samples. So this is the first step that we call data exploration. Now we will know what are the samples we have. We can see that here we have 12 samples, 6 of them being normal and 6 of them being cancer. And you can see that we have only 6 biological samples, 6 humans, 6 people who provided samples. You can see that for each one of them we have 2. One is cancer, the one's normal. So what we need to do now is to go and look at the raw data. The raw data will be just a number of Fastq files. Each one of the data set, each one of the samples that we have in our map will have a corresponding Fastq files. Now, the second step usually that we do is to go in, so we are here. So the first thing that we do is to pre-process our data. Pre-processing, like I said yesterday, includes 2 things. Is to check for quality, that is one, and the second is to remove the host. In this case, the host is a human, and I'm going to increase the size so you can see easily the command. So this is the command that I used for one sample. So this is an example of a sample, which is one of those in the data set. So once you copy and paste this in the shell, it gives you your clean data. Now, 2 things. The quality control requires you to observe the quality and do something if the quality is bad. Sometimes when you do lots of samples, you don't have time or patience to look at each sample individually. So sometimes what you do is to just give a command to the computer and tell it, so if some of my samples are below this quality, trim them, or otherwise don't do that. So there is a wrapper here. It's another tool. All these tools I'm talking about, I will send you a link, or in the final file that I will share with you. There will be a link and a tutorial how to install them and how to use them. So this tool here, this command here, is doing two things, and I will explain what these are. The first is that you give it your input. So you give it, for example, here we give it two samples. I mean, two Fastq files. One is the forward, the other one is the reverse. These are the forward and reverse files of the same sample. Because we use the Illumina, which sequences each read twice to have mate, what we call mate information. Then you say, I want to remove here, you say the database, I want to remove all the reads which will align to the human. So this is a human, it's a human database, it's a human genome into which you have transformed into a database. You may want also to remove Fastq as well. In fact, this is not the actual command. Then I will show you the actual command I ran, because there are a few things you need to consider depending on how your computer is set up. Now, so you align all your data to the database, but before you do that, you tell the computer, the program, to use a tool, Thermomatic, which we talked about yesterday, that is used to remove all the sequences which are below a particular quality. Then how you do Thermomatic, and then you tell the command the place of your computer where the Thermomatic is installed. Usually when you install it, it is clear. Sometimes you don't have to do it. Sometimes you just say Thermomatic, because the computer can look it for that. The command can look it automatically itself. Now, the most important things here are this T4. Actually, this is not very important. It's rather this one. So what you do, and this is tricky. I think one of the participants asked the question, if this filtering doesn't really affect the output, it can be possible depending on how good or how bad your data are. So this word here, sliding window 420, this means that you want to remove all the sequences which have quality score below 20. And 4 means that your computer doesn't remove one basis or one at a time. It looks at 4. It looks at 4 bases at once. If the average quality of those 4 is below 20, it removes those 4. Then it continues until it reaches the 4 first bases with the quality that equals or is above 20. Then min-len means that, so let's say you have your sequences, your reads. Your reads are maybe 200 base pairs if you have sequence with maybe a MySeq. But if you sequence with the next seq, the maximum will always be 150. So what is here min length? This means minimum length. So if you have removed the sequences, if you keep removing sequences of bad quality, then you exceed 50% of the length of the read, you tell the computer to remove it. So that means that the minimum length of the remaining good quality will have to be at least half of the original read. Now, this particular tool uses bowtie to remove to align your raw read to the database, in this case to the human genome. And the bowtie is a short read aligner there are others like BWA. If you are used to using graphical user interfaces, they will usually ask you to choose Bowtie or BWA. There are others, but those two are the most common, Bowtie and BWA. So that was the means. Then these are the options of Bowtie. So Dovetail will tell you to remove those reads which align inconsistently. That's important. So now, what I put in my computer myself, in my own computer, was a little bit different. I will go back to the original. So it was something like this. So I put the data and then told the computer that I want to remove human. That database, I called it human, and then I told it where my Geomatic tool is. So the result of this command is something like this. So it shows you a number of reads. So you see that we have those paired. These are the only reads that we want. All the other reads that I put in here are the reads which either were removed because of the trimming process, or those which were removed because they didn't have matching pairs, or because they simply align to the human. We don't want to use those which align to human because we are looking for only microbial data, microbial reads, sequences. So now, after this command, you will have good quality data, and from which we have removed the host. In another analysis that I will share with you later, I'm using pig. I'm using two samples from two conditions in a pig. So from two different samples in a pig farm. One where the sample was collected on the floor versus when you collect from the rectum to see if the microbial community is the same, and most importantly, to see if the antimicrobial resistance profile is the same in those. Now, the next step is easy, not in terms of what goes behind the scenes, but rather easy on how you type it on the computer. Is now the taxonomic profiling. So after you've done what we say, you will take all the good quality data and then you put them together. Why? Because now we have gone beyond a step where we need that paired end information. So whether we have forward or reverse doesn't mean anything. All we do is just put all of them together. Now, we will do that. When we do that, we run a command, which will be in the file. And what we get after that is something like this. We just get one Fastq file here, and this Fastq file is the only one we will use for the next next steps of the process. Now, when we say taxonomic and functional profiling, this taxonomic profiling is achieved by a tool called the Metaflan. Never mind about the two, as I told you, these two have been recently updated, and it's now Metaflan 3. And for running them, we don't even need to put a number, we just put Metaflan or human, and it does the job. So the Metaflan will usually run the taxonomic analysis, and human runs the functional analysis. So now human, as I told you yesterday, these are the tools which use the classification methods which do not rely on assembly. So which means these tools will not do the assembly or binning. They will just run the classification based on a sequence, also based on the composition. The good thing is that when you run the functional analysis human, it automatically runs the taxonomic analysis at the same time. And this is the good thing with the human, because although it doesn't do the assembly, it tries to link the functions to the taxons, which is not the case for some of the tools. And then this is the command to run it. It's easy to run. You just type human threads. The word threads here means if you have a computer which can run parallel, which can do the same job in what we call threads, it increases the speed and also the capacity. But this requires a high capacity computer. Most computers that people use in our homes, like the computer will have they will be able to run at least 4 threads. You can put a number here depending on how processors your computer has. And then you put it the reads, the ones I just showed you, we got right after the pre-processing. Then you say how you want to call the folder into which the results will be put. Just that's it. Now this I wouldn't run this right now, because this when I ran it yesterday, it took like 3 hours to complete. So what I will just show you is what comes out of the process. Again, I will go to the file, to the folder, and comes here. So as I told you, it runs Metafran and the human, and into Metafran it gives a number of tables. Now these tables will open one of them. They include just this one. Yeah, yes. It includes a list. See if I can increase the size. So it takes the taxons, and the taxons sometimes you see they can, depending on how the read was, it can just show you that it's bacteria. In that case, we don't know what bacteria those are. They can go up to just what is this? These are orders, the families, I guess. We know that these are firmicutes or proteobacteria, but we don't know the genus. They can go up to the possibility of you to know what species these are. Some of the species. But it tells you now each taxon, whether it's a complete taxonomic entity like species or genus, it will just give you the number of those that align to 8. That's what we get from this file. Now, the next thing is, so once you have these files, so this is what you get, for example, here after Metafran, it shows you the percentage of the reads which mapped to this taxon. And then what you do after this step is not different from what Dr. Mira was explaining. It's just now the visualization and the statistics to look at the meaning of that. So I will show you an example on maybe one of them. We'll use the same software Mira talked about. It's Ara Studio. So I don't want to just do a lot of coding. I will just try to make it as if we're using a graphical user interface. To run the graphical user interface we will use, you you type a simple command. Yeah, it's called Pavian. You just copy and paste this line here. One computer using a bad bad combination because I've changed the computer. I don't know why they don't use the same commands in all of them. Okay, so here we have it. Now, so it opens a sort of a graphical user interface that you can use for analysis. And then it will ask us to put in the data. Let's go, for example, and take, I'm sorry. Yeah, it's there. Then we put in, we can choose to put all of them, but I will put only like 3. I will just put 2 samples just for simplicity. Now, once you do that, it tells you that you have uploaded this number of files. So we have uploaded here 4 files for samples, and it tells us that the report, the 4 valid reports in total. This is because we didn't have any problem uploading them. If there is a problem in uploading some of them, there will you may find that the number of files and the number of valid reports are not the same. That will be mean that one of the files is not correct. So once we here, then what we do is to go to the results overview, and the results summary tells us that we have the number of samples are here, and then we have the number of reads. These are percentage of all the reads which are classified 100%, and most importantly, all our reads are from microbial reads, and almost all of them are bacteria, with an exception of one which has like a few reads which are viruses here, which are virus reads. So this means our 2 processing programs, which are Metafran and human, and in this case Metafran, did a good job of eliminating eukaryotic reads, especially the host, which is human. Now, we go to the samples now. The samples will show the sample figure. It's called the Sankey visualization, will show us the composition in these samples, what we have. So we have all of them here in this sample are bacteria, because we can change which sample we're looking at. So this particular sample has all of them are bacteria, and they tell you how many, what is the percentage of those are actinobacteria, and until the level we get to the species. You can see that some of these, like this Cytophaga, is a genus, but then we don't have the species for those ones. But we have species for a couple of them, and we can do the same by changing the sample we're looking at. We can also do it by looking at a table, taking one of these and see what are the other things we have. I'm curious to see, for example, if I put this and I go to the key. I think there's one of them that had that viruses, but I can't remember which one it was. Yeah, it's this one. You see, it shows it's a lymphocryptovirus, which is not classified. But this is what we get usually when you have a sample that has both bacteria and viruses. And another thing here is that we are looking only at a couple of them because, as I told you, this is a subsample. It's not the entire data set. since it's not the entire data set, that means we have a lot of things that we are not able to see here because most of the data, this was like only 1/10, not even 1/10 of the actual data. Then another thing you can do is the comparison here. So when you do a comparison here, you can choose to see what do you want to compare. For example, here I would say that I want to just look at the species. In that case, we get only those taxa that have been identified to the species level, and then we see how they're different in different how they differ in different samples. So, for example, in this case, we can see that we have a couple of bacteria, Capnophaga, which was quite unclassified. The one we saw it's unclassified, but we don't have it in these two samples in this individual P140. But we have it in this, and then if we take maybe one here, Prevotella, we have it in this individual with cancer, but we don't have it in the individual with normal. But again, as I said, the fact that we don't have it doesn't mean that it's not there. It could be that the data we have, because we have reduced the amount of data, doesn't show exactly the full image of what we should have. Now, here I'm not so. The same thing we did here for taxonomic classification is exactly the same thing that we do for function classification. The only things that the change here, instead of having the names of species, the name of taxa, you have the names of a gene. Maybe it could be the names of genes that we have, and see how those genes are distributed in the samples. So at the end of the day, what you do, you can now download the table. And when you download this table, it gives you just the tables with the samples and then all these taxa, and you can use it to make just a normal, like what you do when you want to make any visualization figure. You can even you can do it better in R because it has more visualization capacity. But in this case, you can even use simple Excel spreadsheet if it's not too many taxa to give you a stacked bar chart. So the end point would be to have something like this, which we could reproduce easily if we, using the same process, if we use the entire data set. So this is the end point at this level, not the end point, but the first step in data analytics and the data interpretation. So you see we have a couple of samples here they're showing only 5 samples, although we had 6, but the same. So what we see here is that we have a distribution of our taxa, and because some of the taxa were not identified to the species level, they just want to have only the families. And for example, you can see that for just a simple observation, we have more bacteria in samples with cancer as compared to samples without cancer. So we have, I think, more firmicutes in normal versus in comparison to cancer. Now, the next step would be, of course, which is not what we will do today, to put the data into statistical software like DSEC and others, which will tell you whether there is a significant difference. Because here, you can't just say, I'm looking at this and it's obvious that there's more of firmicutes in normal as compared to cancer. So there have to be proof, statistical proof of that significance. So so I will not we'll just end here. As I told you again, I will share with you all these steps for a particular data set, which I will show you how to get it. And then I will show you step by step on how you can set up your computer to run these particular commands. But that's one aspect, because all the commands that we've been running are on the command line. There is a software tool that I want to share with you, especially for those who are not comfortable with the command line tools. It's called Megan. It's one of those we presented yesterday. Megan 6. Currently, they are on version 6 of this tool. Now, I will share with you this link. This is the link where you can access Megan 6. And again, there will be clear instructions. It's almost written. So the Megan 6 has 2 versions. They have a Megan 6 Community Edition. So this is a free version. You don't have to pay any money to use it. And it has 3 executables. So what you do is just download the one that depends on your computer. So if you're using Windows, you download the one that ends with XE. If you're using a Unix computer, especially if you're using Linux, Ubuntu, or any other Linux computer, you have to use the second one, which is it's a shell. Then it's a bash file, or Mac. Now, I would, at the beginning, I would urge you to use the Megan 6 Ultimate Edition. This one is not free. You have to pay for it, but they give you 30 days of trial, and you have full option. Like right now, I didn't buy it because it costs almost $3,000 per year, which I don't have. But I'm using it for these 30 days. Just for this workshop, because I don't usually use this software. I use the command line software, but I think it's a good one. And also, the good thing with this Megan Ultimate Edition, the full option, it's because it has more, it comes with more databases for functional analysis. So the free option doesn't have this KEGG function database. But the full option has them. This is the most popular, it's the Japanese database, which is really very complete. The only problem is that now they have made it, you have to pay for it. I think that's the reason why this Megan Ultimate is now a paid service, because they have to use this KEGG, which is now you have to pay to use it. But for the first 30 days, you can just use it. And then when you have this Megan 6 Ultimate, you can also download this taxonomy file, because this taxonomy file has all the taxonomy information, all the functional information that you will need to use with the Megan information. So what you have is now the final once you download it, they will tell you where to put the license. They'll show you where to put the license, and when you download it, they send you an email with a line that you copy and paste into the license window and to be activated for the 30 days. For example, I'm going to use it, show you how it looks like. It looks like this. Okay, so this is how it looks. So what you do when you want to analyze it, the only problem with this bioinformatics, sometimes it's difficult to do everything by visual graphic interface. So the first thing is that you take your Fastq file, the one you have cleaned, then you change it into a FASTA file. There are tools, some of the tools are even free or online, that you can put a Fastq file, and then they make it a fast file, which you can now blast. And then what you put here is the table that you get out of the blast. You just use BLAST because BLAST has all the taxonomic information. Then you just go to the file and then import your taxonomic file. Your file that you want to use. For example, here you can import one of the things we have been working with. Then it will immediately, now you change, you tell the software which classification you want to use. You say, I want to do taxonomy, and I also want to do maybe a functional analysis with Cake database. Then you just apply immediately. It will start running and tell you do the same thing that what we just received from Metafran, we just did online. We did on the command line. So the only things that this particular software right now cannot work on this particular computer, again, because I don't have all the setups. Especially it uses what we call a Java environment, and I don't have it on my Mac because I don't usually use this for analysis. But I couldn't get the other computer hooked to the WebEx. So yesterday I tried again to use, you can try this, I think, MGRust. So you can use this. The good thing with this MGRust is that after you've cleaned, you have a clean Fastq file, you can now put it into MGRust web tool. You click on upload, and then you upload your data. But with MGRust, the reason why here right now it doesn't accept is because before you use Rust, you have to register. And when you click on register, it sends your information to a person who will judge whether they want to give you access to Rust or not. So that's why some of these tools are not ideal, because you are using other people's resources. Sometimes they will restrict what you can do. For example, here I have requested access to this for like twice, but until now I haven't heard from them. And also, even if they give you access to it, sometimes you upload your data, but then you have to wait for a particular time, a certain length of time over which you don't have any command. And also it's unpredictable. Sometimes they give you access immediately, sometimes you have to wait a couple of days. So it's not ideal, but if it's the only option you have, you can explore it, especially if you cannot run other software like Megan. But I think, in my opinion, Megan, the one I just showed you, is one of the best because it's a graphical user interface. The only things that you need to just do the preliminary data, like the cleaning, the trimming, and the removing the host outside using other tools, the ones I just showed you. So I will share with you all these links, and also a step-by-step tutorial, which is almost done because I'm waiting for one analysis to complete on my other server. And then I will share with you, with Dr. Zende, we will share with everybody else by the end of the day today or tomorrow morning. I think it will be tomorrow morning in India because you are a couple of hours ahead of me. In 3 hours, yeah. Thank you. If you have any questions, I will be happy to answer some of them. So maybe before I leave, so there's this website, just to have an idea, because I think yesterday there was a question about how much money you would expect to spend for a metagenomic project. So this is a service from some laboratory in Canada. So all the dollars, all the money here is in Canadian dollars. So you can, depending on what you want, you can have an estimation of how much you would spend. in a lab. For example, if you want to do an amplicon sequencing versus when you want to do metagenomic sequencing. So metagenomic sequence, here they're referring to the shotgun metagenomics. Again, this doesn't clearly distinguish those two, but they're not. It's a part of it. So metagenomic sequencing, here we are looking at the high depth and the regular depth, but more importantly, we'll be looking at the high depth, because if you have only 1.2 gigabytes per sample, that would be very, very low. So we are looking at these two last options where you have 120 or even more, especially if you want to do the assembly. Now, depending on how many samples, they tell you if for them, if for let's say, for example, you have 10 samples, it's 9 to 10 samples. If it's an academic institution, but this is usually if it's like a Canadian government, we can do an estimation around maybe $10,000. But this is in Canada. In India, it might be a little bit higher because of the cost of some of the reagents. But a decent metagenomic project will be around that amount. So something between $9,000 and $15,000. So if there is any question, I will be happy to answer.
Thank you, Morris. Thank you for that awesome presentation. So let's open up the symposium for question answers. We have Morris and we have Mira. Feel free to ask the questions, or you can type it in the chat box.
Hello, Dr. Bhushan.
Yes, Bhaskar. Yeah, go ahead.
Meanwhile, the people, they are phrasing their questions. I have a little query, maybe Mira or Dr. Morris both can answer. What is the greatest challenge what you foresee in the genome sequencing?
Morris, Mira?
So you mean the challenges in genomic in general, or particular specific for metagenomics, or in general?
Yeah, what the greatest challenge what you foresee for metagenomics?
Okay, perfect. So for me, I see two things. So these days, from a bioinformatics a point of view, one of the challenges that when people have access to things, they tend to sequence. So for example, now I was looking at one of the data set to use for tutorial, and then there was one particular data set in NCBI, and I downloaded it and then started removing all the hosts. And when I removed the host, there were only two reads were aligning to the microbes. So what that means is that sometimes there's a disconnect between the labs, the demanding labs, and the analysis part, because you think that you have a sequencer, then you can sequence things. But if you don't plan, like we said yesterday, and know how much depth, how much data you need to sequence, and then you will be losing your money, because you use all the money to do the library, to buy sequencing kit, and at the end of the day, your data is useless because you have sequenced almost just human or just a pig. So one of the things which is challenging too is that there has to be an understanding of how bioinformatics works. So how much should we sequence? So if you are a lab, you want to sequence to learn to answer a particular questions to do a particular investigations, you need to consult with bioinformaticians first and say, okay, this is how much you will need. This is how much data you will need for me to give you a meaningful analysis. So that's for me the major challenge, because all the work that is put into the bench work is hard work, and it's also expensive. So it's a shame when people spend that resources and at the end of the day they don't get any results.
Just to add on to that, as we discussed yesterday, everything starts on the question. And I think we need skilled and trained personals in different phases of this metagenomic projects, because we need to know what we are putting in. As Dr. Bhushan mentioned earlier, we need a skilled person to know that what we are putting in, we have good quality DNA, we have all the things to tell that our input is good to start with, and whatever we are getting output, we should be in a better position to interrupt in a way that it's without any much biases. So one of the challenges is all of us can do sequencing, but the thing is, are we doing it the right and proper way? Do we have the skills and training to get this money going not into waste, but of meaningful answers to the question we ask? So definitely train people and get them trained and do good research projects.
Yeah.
Good point, Meera. Good point. Very good point. Yep.
Yeah, thank you, both of you. Yeah, definitely it is going to realize the entire, you can say, the whatever our understanding about the microbial biota and everything. Thanks, both of you. Anyone else from the audience, please? They can shoot their questions. Any queries?
Yeah, ma'am, it's a very naive question, but all the same, after hearing the discussion, I would like to ask you this as a total novice. Between doing a lab search, this is just a situation that I'm posing in front of you as a microbiologist. Say, if one would be looking at the interaction of a particular drug with a population in a normal and a disease state, would it be smarter to use a bioinformatics approach as to a lab-based approach? I mean, an experimental approach. Which would you recommend?
Both has its own pros and cons, I would say, Meera. So if you want a bigger question answered, it's like you can do the sequencing and do bioinformatics approach, right? And then you will get a lot of information. Then if you want to pinpoint something, you can go back and do a lab experiment. For example, there were studies, I would say, I was so fascinated about some of the studies done in the microbiome field, where they look into what is happening in a global perspective inside the gut when you give drugs, right? And when we get into this metagenomic sequencing and we get into a group of bacteria, or let's say a function, then we can go back to the lab and see whether we can culture them, right? If it's unculturable, that's no point. But if it's something we can do to culture these bacteria and see this effect on lab-based effect, then we are proving it at whatever we see on a global perspective. We pinpoint it to this bacteria or virus, and we are trying to see the direct interactions with them. So both go hand by hand. Again, it depends upon how much you can put towards it. If you want a global answer, first go with the sequencing approach and get the answer and pinpoint to the question which you want to. That's what I would say. But I would open it up to Dr. Bhushan and Dr. Morris to their opinions as well.
So great start, Meera. So Dr. Shankar Kuti, can you please tell me what actually project are you doing so that we can tell you what to use based on the knowledge that we have? So if you can describe your project, that'll help Morris, Meera, and myself to kind of